Okay, I think we are live, but let's wait a few minutes just to double check everything. Okay, okay, we are live. Good. I guess on YouTube it looks also okay. Oops. Okay, so then let's get started with our third lecture. Welcome everyone. Today we will cover basically the ways that we convert the biological molecule or DNA or RNA to the digital data so that we can analyze it. With that, I'll start with some reminder slides from the previous week. We covered some of these. So remember, we have several steps to generate the digital data for analysis. And we usually start with sample collection, where we get the sample from an organism. This can be blood, cell, tissue, anything. And then we collect the sample, and then we prepare the library. The library would be an intermediate step so that DNA can be read, can be sequenced in DNA sequencing machines, for example, so that they can connect to the corresponding parts in the DNA sequencers, so that we can start reading, so that these machines can start reading these DNA. This is a necessary step, perhaps maybe for now, for the sequencing machines. And then eventually what we get is the chopped DNA fragments. These chopped DNA fragments are either due to the limitations of the sequencing machines or simply because of the physical factors, which makes it very hard to keep the DNA in its original structure, and perhaps in the chromosome size. They will get fragmented essentially as you even move these fragments. Then we have this raw data that is generated by sequencing machines based on how these machines are designed, based on the technology that these machines are using. Some machines will generate the image data, some machines will generate raw electrical signal, and this is simply because how these machines are operating are at the lower level. Then we will analyze this raw data, usually by first translating this raw data to ATGCs using a computation step called base calling. And then the next steps will use this base called data to do the analysis. But there are also some other computational approaches that can directly analyze this raw data. And almost all of the computational steps essentially lie in between these two regions. So this is the pipeline that we showed earlier, and we essentially stated that we will be covering almost all the steps here in the pipeline throughout this course. And we will start with this step, which is converting the DNA molecule to the digital format so that we can analyze it. So then let's start with a high-level picture of how this comparison looks like. So we have these high-throughput sequencing machines. So these are machines that can look like this, or there are other types of machines. But essentially what they do is they take a biological molecule, maybe a DNA or RNA, as input. And as output, they will generate fragments of DNA sequences that we call reads. So we're going to be using the term read a lot throughout this course. And when we say a read is essentially a fragment of the DNA. And that fragment is a read, and then that is read or sequenced by a sequencing machine, and then corresponding raw data of that read is generated, but that raw data is still a read. And then you can base call that read, and then a base called version of that read is generated. But the base called version of it is also still called a read. So to simplify it in your mind, you can just think of it as the fragmented molecule that is read by a sequencer as one piece. So that is a read, essentially. I'm clarifying this because I've got several questions in the past about this, and it's not necessarily clear in the field, and it's not necessarily also clarified in the field to explain what it is. And essentially there are certain challenges to using these sequencing machines. One, when we generate these reads, we will lose their origin information. We don't know basically where they are coming from within the genome. Second, these sequences, once they are converted into their digital form, they may contain errors. So these errors may be introduced by the sequencing machines itself due to the noise or the technological limitations that they have, or the later computational parts, for example, that translate this raw data to the individual bases. And as a third challenge, specifically perhaps a computational challenge in the practical sense, is we have many of these fragments to analyze. So assume we want to analyze a human genome, and one pair of the chromosomes, considering one pair of chromosomes, we have 23 pairs, right? And in total, we will have 3.2 billion bases. If you consider both parents diploid genome, we would have around 6 to 6.5 billion bases within a single cell in the human genome. It is not necessarily we are just sequencing the entire genome just once. We are sequencing it multiple times. It's done by either copying the same information multiple times with amplification, or essentially using multiple cells. But essentially what we will generate is usually a multi-copy information of your genome. And assume we cover your genome 10 times, meaning from a single parent, we generate bases of 30 billion bases. Then this means that we have a 10x of sequencing depth of coverage. So this coverage is needed because a single read may contain error, or we may not be able to analyze that read correctly. But having multiple copies for the same region in the genome sort of enables us to make a more accurate decisions, more accurate analysis. So that's why we have a multiple sequencing depth of coverage. So that's why we have many also sequences to analyze, because one copy for now is not enough. But essentially, as you might have observed, DNA sequencer is a chopper. We start with a large DNA molecule, maybe it's your entire chromosome. This is chopped into small DNA fragments, which are reads. And then the digital format of these reads are generated through the sequencers. There are many sequencing technologies that are utilized by the companies, for example, such as the Oxford Nanopore technologies utilizes the Nanopore sequencing technology. PacBio uses single molecule real-time type of sequencing, or SMART. Illumina mainly utilizes sequencing by synthesis sequencing technology. So underneath of these companies is a sequencing technology, a sequencing process that enables these companies to develop their own sequencing machines and their own solutions to sequence the DNA. And depending on the technology, the sequencing technology that is used by these companies, a different raw data is generated by them. And we will be looking at these raw data and how they are generated. And we are also interested in understanding these newer sequencing technologies and what we can do with them. And if you're interested in, for example, what we can do with Nanopore sequencing technology, there's a relatively recent paper about this, but we will be covering more about this also in this lecture, and I'll be providing you also with more pointers to papers. So let's start with the Nanopore sequencing. I'll cover in this lecture hopefully also how Illumina works, how sequencing by synthesis works. But I'm starting with Nanopore sequencing because this is something also dear to my heart. But I also believe it has many unique and new opportunities that perhaps we should tackle now. So this is a picture of the main steps in Nanopore sequencing. What I want to here actually show is these Nanopore sequencers, the technology itself is so tiny that you can design a product that is essentially small, that can even fit in the palm of your hand. So this is simply because how essentially each sequencer, each physical structure in these sequencers that can sequence a single DNA, how tiny they are so that they can fit in a very compact form. And then there are, of course, like certain elements here. This is the flow cell. This is a consumable that you, for example, buy and then attach it to your, let's say this device is a portable device called MinION. And then when you prepare your library accordingly, you'll start sequencing your DNA or RNA. But on top of this, there is this sensor chip, including many of these tiny pores that we call nanopores. So we have many of these. This is usually around, we have usually around 500 to 2000 of these pores on a single chip. And so then this means that we are sequencing many DNA, many copies of DNA. This can be either same, which is not necessarily the case, but usually different copies of DNA in parallel at the same time. And if you look more closely to a single pore, what you're going to see is potentially a three-layer solution. We have a top layer, a membrane, and also a bottom layer. I'll explain these actually later on in the later slides. But essentially, what will happen is that we will be measuring the ionic current disruptions on this ion as the DNA moves through this tiny pore, again, that we call nanopore. And these electrical current disruptions are informative for us to sort of sequence every base, to understand each base in the DNA as they move through this pore. But this didn't start as essentially as complicated as this, as the image that I showed earlier. It essentially started with an idea in 1980s by David Deamer and his colleagues. And the idea was, actually, this is his own original handwriting on a notebook. And the idea was basically, it started with asking a question. The question was sort of like, DNA is mainly negatively charged. And can we build a system, can we build a structure such that we can let the DNA move from one direction to another direction? And to do this, one of the good ways is actually to create essentially a voltage difference where you have one layer that is negatively charged and the other layer is positively charged, and then you let the physics work sort of so that a negatively charged DNA also is drawn towards the positively charged side, right? So this is due to the electromagnetic field that is around this region, and then this negatively charged molecule would be literally moving towards the positively charged side. So this was the question: can we move DNA? And the second one is, as we move this DNA, can we are essentially the ionic currents or the charges that we have that is essentially attached to each base in the DNA, can we somehow read them? Can we create an environment so that this DNA, the charges in the DNA, disrupts the ionic current flow around the region where this DNA would be moving, so that this disruption would be informative for us to tell what sort of DNA, what sort of base is moving at that particular time. Would it be creating a descriptive and disruptive change in the environment? And it is descriptive enough for us to say, okay, this is the A that is now moving through that pore, because it is causing a disruptive change and also a descriptive change in the field. So this was the next question. So asking all of the, and this is a physically plausible question. These are physically plausible questions, right? And theoretically, perhaps even correct. And again, this is the question that David Beamer and Deemer and his colleagues were asking. And this is basically what he imagined. But essentially, there were limitations to it. The idea works on the paper, perhaps theoretically, but it didn't work in a practical manner. And the reason for that, the question to answer, can we move the DNA, was yes, we can move it. But at what speed? So earlier, the scientists figured out that this DNA moves at a very high and also at a very non-controllable speed. So when this DNA is moving at a speed that varies a lot and at a speed is very high, then this means that you cannot really measure the ionic current disruptions here such that at a resolution that you can look at essentially the disruption for every base. So imagine your resolution is just so low that you're only, for example, taking a picture for every, maybe taking a picture is not a good elaboration here, because literally we would be taking a picture for another sequencing technology, but just for you to imagine we're taking a sample for every 10th base and so on. So whatever we have in between, we would be losing the information. And again, there is a variance of the speed and so on. So it was essentially challenging early on. And I'll go back to this slide again. And as essentially people worked on this, main development was this essentially the mother protein here that essentially controls the speed that DNA moves through that tiny pore. We were able to reduce the speed, and we were able to somehow control the speed as well. Although there's still a speed variance, now the speed is in a more controllable fashion, where the numbers are usually around 400 to 500 bases, nucleotides per second. And then since we have the sufficient technology now, we can essentially take 5,000 samples per second, the electrical samples per second. So this means that we can actually take 10 samples per base. This means that you have maybe enough information to look at the disruption that each base is causing in these pores. So this was basically the idea in nanopore sequencing, and this is how we improved it over time. And nanopore sequencing is a widely used sequencing technology because of its advantages. For example, we can sequence long reads up to 2 million bases. So we can achieve such, we can sequence such long reads because the only limitation for us not to achieve even longer, not to sequence even longer reads is essentially the tangling issue that the DNA itself, it poses to itself. So as essentially as you go longer with much longer reads, the DNAs can get tangled. So it may block you, it may prevent you from sequencing those reads at all. But other than these, there is essentially there isn't any other major limitation that prevents us from reading much longer reads, compared to other sequencing technologies that I'll try to cover what limits their read lengths in their technology. As I said, this is in a very compact form, so this means that we can design sequencers that are very small. Specifically, this one here is a mini-ion that can fit in palm of your hand. So this means that we can even achieve portable sequencing in the field. So these are relatively cost effective. You can spend around $1,000 to sequence your own genome. And there are also some unique features, for example, real-time analysis that I'll cover next. So this is a bit more lower level look how nanopore sequencing is working. So as I said, imagine a 3-layer solution. This is top layer is negatively charged and the bottom layer is positively charged. And on the middle part, we have a membrane where the nanopore is attached and where essentially the currents are flowing. So we apply a voltage at a certain direction such that we cause this the electromagnetic difference on the bottom and the top layers. So since the DNA is also negatively charged, what's happening is that once the DNA is attached to the pore, with the help of this motor protein, we start sequencing it. And usually it goes with, specifically for the DNA, it usually goes with, you break the double strand structure of the DNA. So now you are essentially sequencing a single strand of the DNA. And as this single strand goes through this pore, the ionic current, the current that flows through this membrane will be disrupted. And these disruptions are informative for us to tell each base, as I told earlier. And this, as a result, we will be outputting ionic current measurements as the DNA passes through these pores. And this is generated at a certain speed, which is, again, as I said, is around 5,000 electrical signals per second, or 400 to 500 bases per second. And what we can do is that once we generate these electrical signals, we can perhaps even convert these electrical signals to ATGCs. This is a step called base calling. We call the bases from the electrical signals. Or we could directly analyze these raw electrical signals without base calling. Another advantage is, since this data is outputted as soon as it is available, in real time, we say, we can also start analyzing this electrical data as soon as it is generated. So this is an analysis, type of analysis that is called real-time analysis, because we're generating the data, we're analyzing the data as it is generated at its data generation speed. What's even more important is by doing the real-time analysis, we can also talk to the sequencers. Since this is real time, since we're not doing this analysis after the sequencing is done, right? So we're doing this analysis in real time as the sequencing is happening. We can talk to the sequencer. We can give feedback to sequencer. For example, if we sort of understand that whatever the read that we're sequencing at a particular time is not interesting for us. For example, we realize that this DNA is somewhat contaminated in our sample. Maybe we're just interested in the human reads, but whatever the read that we're analyzing is not coming from human read that we understand so far. So we can tell the sequencer actually to stop sequencing that read early without fully sequencing it. So this is an approach that is known as adaptive sampling. We are adaptively sampling or adaptively taking a decision to sequence or not sequence that read. And we can send a signal to sequencer to stop sequencing that read. And how it works is essentially not a magic. Assume we send the signal to a sequencer. And then what happens is that for a short fraction of time, the voltage is now applied in the other direction. So now this direction, this side is positively charged and this side is negatively charged. So what's going to happen is that DNA will still move towards the positively charged, which is now the upper part of this region. So in a sense that you will start ejecting this read from this nanopore, and since it won't have the necessary the adapters, the motor proteins, and so on, eventually it won't necessarily attach again to these nanopores. So once you eject, it is safe to assume that, relatively safe to assume that this won't reattach to the nanopores again. And this is basically called adaptive sampling, and this is useful for many reasons. For example, it can reduce the sequencing time and cost, or it can enable you to utilize your resources in a much better way. So you don't waste your resources and you use them for useful purposes. And also the real-time analysis can reduce the overall latency because we can overlap the sequencing time with the analysis. So these are basically the steps in nanopore sequencing. By the way, if you have any questions, I'm taking a look at the chat in the meantime on YouTube. You can ask your questions there, and I'll try to answer them. So this is how we generate raw sequencing data from nanopore sequencing. Raw sequencing data is an electrical signal for us. So then let's look at how actually we analyze this data. So it is usually done by two steps. One, first step is called base calling. The other step is called sort of raw signal analysis by some denoising approaches, but now I'll cover these. So traditional way is to translate or base call these signals to bases before the analysis. And how it works is usually via you generate the electrical signal and then you input these to the base callers. So these base callers are usually in the form of a deep neural network type architectures. They can, of course, contain much more modern also layers such as LSTM, RNNs, CNNs, and so on. But essentially, what we will do, we will input these raw signals to DNN-based base callers, and then they will sort of understand the complex structure of the electrical signal, and then they will extract the nucleotide information from these electrical signals. So it's like a translator for us, right? So I assume these are two different languages, and this is one language that you don't speak, you don't understand. There is a translator in between, and you ask the translator, what is going on here? What is being said? And then the translator will convert the data, the information to the language that you understand, which is in the human readable form, literally. We will then fundamentally ask whether we will need that translator or not, but this is basically the process that is going on. And these base callers are nowadays very accurate. They can generate data that is much less noisy than the raw electrical signals. There are many analysis tools that is developed that uses these base call sequences, because when you are in the domain of base call sequences, now you're sort of creating this abstraction level that maybe you are not assuming, you're not making an assumption about the underlying sequencing technology. Regardless of the sequencing technology, now you have the ATGC information that might be coming from any technology, right? Maybe this is a nice level of abstraction level for certain applications. But essentially, the translator that we're using in between, they are usually very costly operations, and we need to use power-hungry devices such as GPUs to run this base calling step. There are more recent types of work that try to eliminate this translator, which is the base calling, in between, so that we can directly analyze this raw signal to make our analysis. So it is perhaps can be understood as you don't need a translator anymore, because now you learned the language, perhaps, so that you can directly talk to this particular language and then do your analysis. This is relatively better in several ways. For example, now that we don't need this additional step, now the entire analysis can be much more efficient, perhaps with better scalability and portability, because perhaps we would not be needing the GPUs necessarily to do the base calling. And raw signals, the raw electrical signal, retain more information than just individual bases. So this can also be considered as compression, right? So raw electrical signals retain richer information that is beyond bases. So it can also contain information about the structure of the DNA or RNA. And then this is a very useful information, for example, to understand whether the DNA is methylated at a certain position, so that you can even understand whether this is a region that is affecting the gene expressions and so on. Whether we have, for example, a histone around and so on. So this is an important information, and this is essentially a rich information that we are losing unless you design your base colors in a way that also extracts this rich information and then outputs it in the base code data. So yes, this is the question that we ask: do we need a translator to analyze the genomic data? And the answer is not necessarily. So I'm not saying no in a very, let's say, deterministic way, but it is not necessarily, it depends on the application that you're using. And how do we enable analysis from electrical signals? There are several ways, but I'll try to explain you the common steps that are used in these works. So we first go a little bit deeper and then say that the ionic current flow here, the current flow over there, is not disrupted just because of the presence of a single nucleotide. Rather, the DNA, the nanopore itself, is in a shape such that there are multiple nucleotides that is affecting the current flow. And for older versions of nanopore, this is around 6 nucleotides. For the more recent ones, this is around 9 nucleotides. And this is because we have this narrow region here, which is known as sensing head. So in this region, whatever the base that is closest to the region is causing the disruption most. But still, whatever is surrounding this also narrow region is also causing an ionic current disruption. So this is not simply just the collection of the bases that you have in the nanopore, but their order is also important, because you can change the order. So this means that now the base closest to the sensing head will be different, and also the distance of the other bases will also be different to the sensing head, so such that perhaps you will generate a different characteristics of the ionic current disruption. So these are the important factors here. And of course, the speed that DNA moves through this pore is also varies, which we refer to as translocation speed. But essentially, what will happen is that as we sequence, assume that we have an older version of nanopore, as we sequence the 6 nucleotide of DNA portion, we will generate an electrical signal that is representative of these 6 nucleotides. But then the DNA will shift by one base, right? Maybe a new base will come in and another base will come out of this nanopore. And then, maybe just to repeat again, we have these 6 bases, and these are referred to as K-mini nucleotides or K-mers, and these are sequenced at a time. And then as we shift bases, what will happen is that there will be an abrupt change in the signal. But essentially, a signal that we generate while we are sequencing a certain K-mer is known as event. So it corresponds to a segment of the raw signal that is generated while we are sequencing that K-mer. So this is the definition of what an event is. And so essentially, our goal, so this K-mer will correspond to this event will correspond to a particular K-mer. But essentially, our goal is, as we move this DNA, as this DNA moves through this tiny pore, we want to measure the abrupt changes that is caused by the movement of this DNA. And these abrupt changes will hopefully correspond to sequencing of the next k-mer or of a different k-mer. So in a sense, you can sort of segment these events. So you can use some computational tools that identify these abrupt changes in the signals. And then you do some more computation steps. For example, you normalize because you may be generating slightly different signals across different pores or in different times within the same pore, so that the amplitude of the signals are offsetted. But if you normalize, you'll put them back in normal form again, and then you calculate the means and so on. But essentially, what we will have is, for example, a picoampere value that is normalized, that is assigned to an event. And perhaps this value will represent what sort of k-mer that we were sequencing at that time. So eventually, we would be using these numbers instead of these k-mers for our analysis. So this is usually how the raw signal analysis starts, but perhaps we can even design different algorithms to do this analysis differently. So there is one key observation here, though. Assume we are sequencing this k-mer at different times, right? So we have ACT, TGG again. Now we sequenced and then we generated this 2.21 value. Now, maybe in another nanopore or at a different time, we reach to that DNA 6-mer or k-mer again, maybe just because it appears in another region in the DNA. So what will happen is that we will not necessarily generate exactly the same electrical signal. But the key observation is the electrical signal that we are generating will be essentially similar to what we generated earlier. So this is true in most cases. And then we can utilize this information again to do our analysis. And essentially, we are interested in this direction. For example, in this work, what we do is, can we use these electrical signals? Can we eliminate the translation step, which is the base calling step? And can we use the electrical signals to map, to identify the origin of the reads within the genome? To ask the question, where are they coming from within the genome, by sort of measuring the similarities between the genome, for example, a reference genome and the electrical signal that we just generated, and then see which region is the most similar to this electrical region. So this is the question that we ask in RowHash. This was presented last year at ASMB, and also we continuously improve the mechanisms that we propose. For example, in RowHash now, RowHash 2 is recently published, and it improves certain steps in the raw signal analysis to make it more accurate. We also we understand that the raw signal analysis is not currently widely adapted in the field because it's a growing field now, and there is a lack of tools to do the full analysis for the genome. For example, for now, perhaps we cannot even identify the mutations that is present in an individual without base calling, but we want to achieve this, and one way to achieve this is to identify the exact differences, exact nucleotide differences between a read and a reference genome, for example. But you're not base calling the electrical signal. So how do you identify the base difference using the electrical signal? So we developed alignment algorithms that sort of aligns the electrical signal to a reference genome and still identifies these differences. And these differences maybe in the future can be used to identify the mutations without even base calling. So this is what RowAlign does. We also look at how can we design maybe more efficient, faster base callers, what sort of optimizations we can do, maybe a quantization and so on. So this work covers all such optimization opportunities. And we also look at how can we, for example, perform analog computation to do MAC operation, multiply and accumulate operations that are inherent, that is mainly used in, for example, machine learning applications, specifically in DNN-based applications, specific, for example, in base callers, right? And the question was, can we perform this operation in an analog manner so that we can do the computation maybe in a very energy efficient way and maybe in a faster way? And this is what we explore in this work. And what we show is we can do computation inside the memory to do these multiplications so that maybe we can even do base calling. So this is another interesting work. If you are interested, you can take a look at this. And also we look at what happens when we base call and then do the analysis. For example, when we construct the genome, will there be errors? And there will be errors essentially. And the question is, how can we resolve these errors? So in this work, we use a machine learning technique to understand the errors that are still there even after base calling, and then resolve these errors. And this is basically, you can take a look at this if you're interested in that direction. So also, as I said, Nanopore raw signal data retains richer information than just individual bases. If you're interested in what we can do with this type of information, you can take a look at this paper. So now we're done with Nanopore sequencing. In the remaining limited time, I will try to cover the sequencing by synthesis approach that is used by the Illumina sequencing technology. And at the very low level, what you can imagine or what we have is basically, imagine we have a surface, a glass flow surface over there where our nucleotide or our molecule, DNA molecule, is attached somehow to that surface. The attachment is usually actually ensured during the library preparation process, such that actually we have certain nucleotides here. We attach them on purpose so that they bind to the certain regions in this cell surface, because we will have the complementary part of, for example, the initial regions of this DNA. We will already prepare them, and we know how they look like. So then this means that you can synthesize a complementary version of it, and then this can be your adapter perhaps, and then you can attach it to here, so that you make sure this one attaches to those surfaces and in the glass surfaces over there in the Illumina machine. So this is one step of information that I want to introduce. Another step of information that we already talked about is the connection between the nucleotides, right? So A adenine will connect to the thymine T, guanine G will contain the cytosine C. That we know. So that's the second level information that we need to introduce. So knowing this connection information, what we can do is we can literally, as this DNA stands like this, for example, on the glass surface, what we can do is we can release perhaps maybe agents, alcohol, or maybe either only a single T characters or maybe even multiple characters that are floating around the region, right? So imagine you have many T bases, there's a single base that are floating around the region of this DNA. And also we will have an enzyme that we call DNA polymerase that is responsible for essentially the constructing the other strand, copying the one strand of DNA by looking at the other strand, right? So what we will do is, what will happen is that when we release, for example, T in the environment, this T will connect to this surface, to this base that is on the surface. And this is essentially ensured so it doesn't, for example, connect to this A or some other A, because there is basically a particular, essentially the base over there is designed in a way that essentially the connection, the T and A connection should be made over here, because there's a particular chemical connection here, such that it won't happen here, but it will happen here, basically. So we know that chemical connection type, and then that's how we ensure that this T will bind to here. But essentially, what will happen is that as this T binds to this region, we take a picture. This is because as soon as this T binds to this region, it will actually emit light at a certain frequency due to the binding that just happened at this position, at this region. And we can take a picture of this, and then we can understand by looking at the color what sort of base we just successfully were able to bind it to this region, right? And we don't have to know we have an A here, because we know that a T was just connected to this region. So having this information, we can then assume that we can know that there's an A in the corresponding part, right? So this is basically how the sequencing by synthesis works. It's literally we take a picture and then we say there's an A on the other side. And of course, there are like other agents or other, let's say, single bases that are floating around, and then we're not sequencing a single copy of the same DNA. So we have a cluster of the same DNA. So this is a single cluster. Actually, we have different DNAs here, but what we so assume that this, I assume this region, this particular region is a one cluster, and it will contain the exact same information of the DNA. And the reason we do this because this essentially the cycle by cycle process that we're doing, where like every base process that we're doing is not very deterministic. Sometimes essentially we may be essentially attaching more bases than we need, and then this means that we would be emitting a different light and so on. So that's why we need more information for a base, and this is actually what is limiting also the read length of sequencing by synthesis. The reason for that is we cannot basically grow these bases in an unlimited manner. So we can only grow up to a few hundred bases. This is usually around 200 and 300, because as you increase the read length, and again, remember, you have multiple copies of this DNA, right? The exact same copies. Assume initially we start exactly at the same position. So this means that we have T, and this T binds to the exact same position everywhere in the same DNA. So this means that the T that we're emitting is very dense, right? We can very, in a very accurate way, in a confident way, that we can say, okay, there's a T here. But as we move cycle by cycle, so this is literally done cycle by cycle. Once this T is attached here, we will wash all the information that we just released, and also the extra chemical information that was attached to this T is also washed. But also it will contain chemical information such that whatever we release next in the next cycle can only bind if basically this T contains this information. Right? That's why we won't be binding to any other random region, but we will only bind right next to the region, right next to the base that we just were able to bind in the previous cycle. So this is basically how we are growing cycle by cycle. But it may so happen that due to wrong washing, for example, we won't be able to wash all the necessary chemicals, or maybe we will wash just too harshly, so that the synchronization between the duplicate information will be disrupted. So maybe when you reach to 200, sometimes maybe some of the copies will reach to 220, some copies will reach to 180, but maybe most of them will still be around 200. Then this means that you can still take a consensus and then be confident that, okay, we are at the 200 cycle. So there are some information that tells me different color information. Some other information tells me different color information. But most of the time, again, we have a consensus. But as you grow up to 1,000 lengths, then this synchronization will just be gone completely. So this means that you will just generate maybe even random colors at almost random positions, so that you won't be able to tell which base that was just connected at this particular cycle. So that's why, for example, SPS sequencing by synthesis has this read length limitation. Again, I want to remind you that regardless of the sequencing machine, reads still lack the information about their order and location, where they are originated from, right? And then we need to figure that out so that we can analyze the genome. So this means that we need to construct the entire genome from many sequence reads. And then this means that we need to take the next step in the genome analysis pipeline, which is the read mapping step. After we generate the sequencing data, perhaps even we base call them, we need to identify their origin. And then this is done by a step called read mapping. It has many computational steps, as you see here, so that we can make it practical. But this is really like solving life's puzzle from the sequencing output. And this is what we will be covering next week. We will be covering data structures, some steps in read mapping that makes this analysis practical for us. And this is basically where I'll stop. I don't see many questions in the chat. There is one message that says hello, and I say back hello again to that particular person. But other than that, I don't see questions. So maybe I'll wait a few more seconds just in case a question pops up. If not, I'll stop here and I'll continue next week. Okay, I guess there are no questions. Then thanks everyone, and see you next week.